Thank you very much. So yeah, I'm here presenting on the behalf as a representative of Tomasz Błasiak's lab, we are concerned mainly with in vivo studies. And as I saw the program for this school, I thought that there's this balance between rewards and aversive stimuli in how much we talk about them. So today I thought about showing you a bit of ambiguity in the coding of external stimuli. Namely, what I mean here is aversive stimuli by the reward system. And I'm sure that after yesterday's Wolfram Schultz talk, we are all scientists mainly concerned with dopaminergic neurons. But I want us to be on the same page, so I will show you a little bit about what I'm talking about. So when we think about reward system, we think about midbrain dopaminergic neurons, namely ventral tegmental area, VTA for short, and substantia nigra pars compacta, SNC for short. And there is a whole bunch of information about how those two structures and dopaminergic neurons within those structures are involved in coding of rewards and aversive stimulus, which leads to learning mainly through association as well as reinforcement learning. But this is not only confined to those very, let's say, simple learning mechanisms, as rats are pretty well able to learn more sophisticated skills, such as driving a car. And if you don't know this study, I highly suggest you check it out. It's pretty funny and interesting and thought-provoking as well. Okay, when we think about learning, we think about rewards. And when we think about rewards, we tend to, this is my experience, that we tend to forget that there is a whole bunch of characteristics of reward and any given stimulus, but we only focus on the value of the reward, in this case positive value, which usually drives motivation of the animal to approach certain situations, certain stimuli. But as there is also a quality in sensory information which helps animal to identify objects, to just note if this is something important, this may be something important. And here I'm talking about alerting function of dopaminergic neurons. This is something that recently started to come to the surface, that those neurons may code information about alerting events in animals' environment. But also, besides the sensory information and value, information about value, we have a whole bunch of salience coding properties which will drive animals' attention, just boosting cognitive processing and allowing animal to be aroused and attentive. And so yeah, when we think about value, because we can prescribe characteristics of value to every type of stimulus. It may be rewarding stimulus, which will elicit excitation of dopaminergic neurons, but it may also be aversive stimulus with negative value that will elicit inhibition of dopaminergic neurons activity. And also, as I mentioned, those alerting function, this alerting function of neurons, when during different behavioral states, certain sensory stimulus is able to elicit dopaminergic neurons responses. So, for example, click sound during quiet waking will elicit response, will excite the dopaminergic neurons, but the same sound during a task where there is an inaccessible food is just not as important to elicit this type of response. And when we go beyond rewards and value coding and look at the diversity of responses to aversive events, we have classical value coding dopaminergic neurons that are inhibited by aversive stimulus, such as food shock applied to the animal's hind paw or an air puff. And also we have neurons, dopaminergic neurons, that are excited by aversive stimulus. And when we look at cues associated with those stimulus, value coding dopaminergic neurons tend to follow this pretty well-known signal of reward prediction error. However, salience coding dopaminergic neurons just don't care about the value of the cue associated with certain stimulus. They just care if it's important to the animal's well-being, animal's survival, and they will signal that by always increasing their activity in response to that. When it comes to unexpected stimuli, those rewards and air puffs are coded in a pretty much similar manner when it comes to value and salience coding neurons. Just the temporal resolution is a bit different. And when it comes to unpredicted no outcome trials, those value coding neurons will signal no reward or no air puff by either a little dip or a notch in their activity, and salience coding neurons just once again don't care about this type of outcome. It is not important, just nothing happened. There's no saliency in here. Okay, and those two populations of value and salience coding dopaminergic neurons are both functionally and topographically different populations within midbrain, where value coding dopaminergic neurons that are thought to be engaged in seeking behavior, evaluation of stimuli, and in general value learning, tend to be located in ventromedial parts of both VTA and SNC, and they have different target structures, for example, in striatum, compared with neurons coding for salience, which tend to be localized in more dorsolateral parts of both SNC and VTA, and those neurons, I will repeat myself, are thought to be coding for importance and allow for orienting animals' attention and just boost animals' cognitive processing. And as many researchers investigating electrophysiological properties of certain structures, we also used anesthetized animals during our experiments. And one of the anesthetics that is commonly used is urethane. And urethane anesthesia is pretty interesting from my point of view because it evokes two brain states that alternate in a spontaneous manner, and they are pretty stable over time and highly predictable. And as you can see on this sonogram here, when the dominant frequency at given time is color-coded, so the greener the color is, the more dominant the frequency is at given time, that we have those two brain states. One of them is called cortical activation, visible on the sonogram here, and during this brain state, low theta frequencies may be observed in the signal recorded from, for example, cortex or hippocampus. And this brain state, when we try to translate it into freely moving animals, we can see such frequencies during attentive immobile states, such as freezing behavior, when the animal is waiting for something to happen. And the second brain state is slow wave activity, and during this brain state, slow waves dominate animals' EKG recording, and it is mostly connected with low arousal states such as non-REM phase of natural sleep or just very deep relaxation states. So from the previous study of our group, Magda and Tomasz work, when they recorded local field potential from the hippocampus and monitored cortical activation and slow wave activity, while simultaneously they recorded single dopaminergic neurons basal level of activity from VTA and SNC. We know that basal activity pattern and level both depend on brain state under urethane anesthesia. So during slow wave activity, midbrain dopaminergic neurons tend to burst more, so the level of activity is generally higher compared with cortical activation, when those neurons are a bit slower and tend to be non-bursting, more regular. So given that knowledge, we asked ourselves whether responses of midbrain dopaminergic neurons to aversive stimulus may differ between those two brain states as well. And what we originally thought was that they will differ in some kind of parameters, such as latency of response, duration, or even an amplitude. So to answer this question, we performed experiments very much similar to those performed by Magda and Tomasz, and I recorded local field potential from hippocampus, monitored cortical activation and slow wave activity, and recorded single dopaminergic neurons activity and responses to aversive stimulus, which in this case was electrical foot shock, during those two brain states. So when we go into the results, we've recorded neurons, majority of neurons that we recorded was inhibited by aversive stimulus during both cortical activation and slow wave activity. And we would say that this is something that was previously described in literature. We would call them value coding dopaminergic neurons. We've also recorded a population that was excited by aversive stimulus, and we would call them salience coding dopaminergic neurons. This is not something that we would call groundbreaking. And also a few unresponsive cells. However, what we did not expect was to record a pretty large population of neurons that we labeled switching dopaminergic neurons, and on this example you can see why we labeled them as such. So this is an example of one neuron that during cortical activation was inhibited by electrical foot shock, but as soon as the brain state changed into slow wave activity, the same neuron switched the type of response into excitatory one. And majority of recorded switching dopaminergic neurons followed that pattern of switch. However, we recorded few different types of switches as well, but those are just single cases. And this is something that was not previously described in literature. We really don't know what the possible function may be. As I mentioned before, when we thought about differences between two brain states, we thought about parameters of response, amplitude of response, and latency, duration. We did not expect that those neurons will change the type of response entirely. So we, for now, labeled them as what coding dopaminergic neurons, because we just really don't know. When we look at the level of a whole population of those recorded neurons, this brain state dependency in the switch in response can be visible as well. So during cortical activation, a majority of neurons is inhibited by electrical foot shock, and only a small fraction of them is excited by it. This may be also appreciated on this. Here you can see the response presented as normalized change in firing rate. So you can see this a pretty big dip in dopaminergic neurons response, and also proportion of neurons inhibited, excited, and unresponsive during cortical activation. However, when we look at the activity, at the responses to electrical foot shock during slow wave activity, those proportion changes, and more neurons are excited by electrical foot shock, and this mean firing rate during the stimulus application is not just this big dip, but it is something much more complex. We have a population that is excited, we have population that is inhibited, and the relative response is pretty neutral. However, we cannot say that this is the dopaminergic, that this translates into dopaminergic signal, as those different populations may innervate different parts of, for example, striatum. So the dopaminergic signal will be different in every brain part. And we've also looked at the responses of non-dopaminergic VTA and SNC neurons to the electrical food shock. And once again, those numbers are pretty low in my opinion, but we see those switching dopaminergic neurons here as well. And once again, the type of switch of the response is the same as in the case of dopaminergic neurons, so inhibited during cortical activation and excited during slow wave activity. And if we assume that those non-dopaminergic neurons are GABAergic interneurons, and this is very bold assumption, I must say, then this is something that is also not expected. We would expect to see a complementary responses in that case. However, there is also a possibility that those non-dopaminergic neurons are GABAergic projecting neurons or just glutamatergic neurons. And on the level of population, it is once again pretty nicely visible, this switch in the proportion of neurons responding in different type of way. So the first question we asked was why such responses have not been observed so far. And when we looked into literature, we saw that previous researchers that were working on anesthetized animals supplemented urethane anesthesia with ketamine and xylazine. And when we did so, this supplementation evoked very stable, long-lasting slow-wave activity. So it's the simplest explanation that previous researchers were just not able to observe cyclic brain states. So they just got themselves rid of the opportunity to record what we recorded. And when we look at responses of dopaminergic neuron, this is the one single cell over this period of time to electrical food shock, we labeled this neuron as switching neuron during slow wave activity, and the cell was excited, and during cortical activation it was unresponsive to electrical food shock. During the slow wave activity evoked by ketamine and xylazine supplementation, the neuron was excited by aversive stimulus, which is something that we would expect given that it was excited during slow wave activity before. However, when ketamine and xylazine waned and those cyclic brain state alternations came back, the same cell was excited during both cortical activation and slow wave activity. So this is another argument that such supplementation not only abolishes cyclic brain state alternations, but even after they are present once again, the activity of neurons is altered enough to preclude observations of such population as we observed. And of course, we have further questions. And as I mentioned at the beginning, based on location within midbrain of different neurons, we can speculate about the possible outputs. So we looked more into distribution of our switching dopaminergic neurons, and we see no tendency for them to be located in any particular place within the structures. They seem to be evenly distributed. However, when you look at those coronal sections here, you can see that we don't have enough recordings from those more medial parts. So we just shouldn't speculate about where those neurons may or may not be located. So we just took a guess about possible input to those switching dopaminergic neurons. And focused our attention on so-called aversion hotspots, which is in this case lateral habenula, also known as the anterward system, and relay station between lateral habenula and midbrain dopaminergic neurons, rostromedial tegmental nucleus. And they are pretty well known to be engaged in coding for both rewards and aversive stimuli, as well as coding for salience and value. So we investigated lateral habenula more. So once again, we monitored local field potentials from hippocampus, monitored slow wave activity and cortical activation, and recorded activity of lateral habenula neurons and their responses to electrical foot shock. And once again, we see switching dopaminergic neurons. At this point, it was something that we would expect. But what we did not expect is that we see much the picture that we see is much more complicated than in the case of midbrain dopaminergic neurons, where here we have three main types of switching dopaminergic neurons. In the case of lateral habenula switching neurons, when in the case of VTA and SNC dopaminergic neurons, we had only one main, very visible type of response. However, when we look at the whole population, once again, we see that this shift of response is the same as previously observed. So during cortical activation, lateral habenula neurons are, let's say, less excited than during slow wave activity. And here you can see the amplitude of this response is different between those two brain states, as well as sheer numbers of responses. So to sum up the results, we have the same type of switching neurons in both lateral habenula, in both non-dopaminergic midbrain neurons, and in dopaminergic midbrain neurons. So once again, we end up in the place where we started. We just don't know what is really going on, but we lack in our observations pretty important structure, which is RMTG, and maybe the same direction of those switches may be somehow related to and converted into different signal in the RMTG. So as long as we don't know what is going on between those structures, those populations, we cannot really say we cannot speculate about possible function, but I will try to do so regardless. So we hypothesize, and it is really high speculation, that those neurons, those switching neurons in every structure that I showed you may be involved in some kind of dual coding, and they are able to code both value and salience depending on the general brain state at given time. And it would make sense somehow that during states characterized by cortical activation, attentive immobile states, when the animal has to process what is happening, those neurons will support coding for negative value and support learning of avoidance behaviors. However, when animal is deep asleep or just highly relaxed, the same population will code for salience and will support just general increase of alertness and arousal, which may just wake animal up, draw its attention towards what is happening. And this is not something that is completely new, because we know that the dopaminergic signal depends on environmental context. And this is something that I spoiled you a bit at the beginning, because dopaminergic signal, dopaminergic response to the click sound depends on the context. In case of alerting dopaminergic neurons, click sound during quiet waking will elicit response of dopaminergic neuron, but the same click sound during when animal is occupied with something much more important will just be neutral, will not draw animal's attention. And also we know that when animal is pre-fed on the pellets that were previously as rewarding as sucrose water, the pellets just won't be as rewarding as they were before, and vice versa with sucrose. And we have many examples of coding depending on the environmental context. I just don't want to go into details right now, just want to signal you that. And just to sum up this part, environmental context influences how animals will respond to stimuli and learn. And those brain state dependent responses which we observe under anesthesia, of course, may be in some parts translated into some type of environmental context as well. And this phenomenon is not only confined to the reward system. This is something that we can observe, for example, in the subcortical visual system. This is a study by one of my colleagues from our department, and she shone a light on animals' retina and recorded responses in different structures of subcortical visual system. And among many very stable responses, she also observed state-dependent responses to this light shown on the retina. For example, during slow wave activity, neurons in DLG were not responsive to the pulse of light. However, during cortical activation, they started to code information about light. So yes, this may be connected, at least in some part, with alerting signals. And here I mean alerting signals in general, in the whole brain, not only in the dopaminergic system. And here, urethane anesthesia influences much more than just processing of the external stimuli. This is also something that I mentioned at the beginning, that we know that dopaminergic neurons have different activity level during different brain states under urethane anesthesia, and the same applies to neurons in pontine nuclei. And also something that we would really not expect is that depending on the brain state, neurons in nucleus incertus respond differently to optogenetical stimulation of medial septum, which is just, what is important here is that brain state is able to alter response to very artificial kind of stimulation, which is something that we would not usually take into account. And also it is able to impact the very basic thing that we work on, which is spike sorting. And the type of anesthesia that we use may influence how well the recorded neurons are sorted further. So yes, to sum up, information processing is not simply a passive detection of stimuli by the nervous system. It is highly integrative process. Also, it depends on the context, which here I understand as an environmental state, which may evoke, may modulate and influence internal animal states, namely brain states, which will modulate animals' responses to different environmental stimuli, and responding differently to what is happening will once again put animal into different environmental context, and the loop is never ending here. So maybe we should, while studying associative learning and reinforcement learning, maybe we should just try to take into account or just remember how dependent on context and environment the dopaminergic coding is. And with this, I would love to thank you for your attention.
Okay, thank you very much for this fascinating and very interesting results. And we have time for 2-3 questions. So, the gentleman in the back.
Thank you for the lecture. I have 2 questions. First of all, do you think there is a physiological switch between the type of input or the processing of the input that's happening in the neuron between the active or the resting states that changes the coding of this signal? Or do you think that in the resting states any type of activity could be coded as a reward? And my second question is sort of related but different. Do you think this could be some sort of attentional modulation, in the sense that when there is active attention, the signal can be processed in a more complex bidirectional way, whether it's a positive or negative value, where in the absence of this attentional modulation or unmodulated, it is more like signal activity. Everything causes a spike in the same neuron. Thank you.
So the simple answer is yes, but more complex answer is that I think it is about input mostly, and that during different brain states, this one of inputs will be switched off, one will be switched on, and that will create those changing responses. But we don't really know. We are basically starting to learn more about those neurons, and right now we can't pinpoint exactly what kind of inputs they may be involved. We tried to do so with lateral habenula, but it told us nothing basically. So I don't think I can answer your question. I could only speculate, and this is high speculation here. But mostly I agree with all of what you said. It can be all of that, it can be none as well. I don't have enough information to speculate about that.
I mean, one design idea could be that the animal is awake and attentive, but in place of a distractor, so that it is not a resting state, whereas the attention is directed elsewhere. So in theory, there shouldn't be attentional modulation on that specific stimulus. But anyway, that would be something.
That would be interesting, and there is a possibility of that.
Thank you.
Thanks.
Okay. Next question. Yeah.
Thank you. Wonderful talk. My question is associated with the effect of ketamine on those switching neurons. So you showed that once the ketamine weans off, the neurons still are in the state of loss of that function of switching. So first, what would be the behavioral consequence of the loss of that switch? And how you showed that this is like prolonged effect. You showed that it lasts even after removal of ketamine, right? So how long does it last?
We don't have information about how long does it last, but when it comes to ketamine, it is used as an antidepressive treatment, acute antidepressive treatment, to block lateral habenula. So we would guess that this inability to come back to the switching types of responses is mainly because we inhibited lateral habenula for a long period of time, and this inhibition could last about 2 to 3 weeks, if I remember from the literature. So during this period of time, based solely on those responses, I would speculate that those neurons will just code information about salience, and they won't support learning about value, about aversive events, but it doesn't mean that the animal won't learn, won't have value coding, because this is just only a fraction of neurons, and those value coding neurons, their activity is not impaired in any way.
Yeah, and why do you think it's 3 weeks? I mean, I know it's 3 weeks, but why it is that time window?
We don't have recordings from single neurons on how long it may last, but we just know from the literature that after this shot of ketamine, it usually lasts about 2 to 3 weeks. So I would just guess that this is how long it will last.
Thank you.
Okay, and then lady here. Can you please raise your hand so we can give you a microphone?
Thank you. So we have our somewhat consistent long-term preferences for any discretized object category or concept, semantic concept. So what's our understanding about how this immediate reward encoding dopaminergic neurons get converted to this reward gets converted to long-term preferences? And what's the memory system where these long-term preferences are stored? Is it same as the semantic memory system, or is there another memory system with 1:1 correspondence for each of these object category and preference?
I don't feel like I can answer that, because we only focused on the very immediate type of response to the stimulus, and what we don't know about what is happening farther down the line. So there's it's a bit awkward to try to answer your questions, because we don't have any more of information about this population. We just somehow stumbled upon it and are left speechless on what it can do. So I don't know.
It might be a totally different kind of reward encoding mechanism.
I would assume so. It may be even more. We know that this is a very heterogeneous place, dopaminergic neurons in the midbrain. So,
okay. Thank you.
Okay, do we have any questions from the audience? Okay, I may have a last question. Do you have an idea about the phenotypes of those switching dopaminergic neurons? You mentioned that you have no idea about the location, or you cannot pinpoint the exact differences in location, but maybe other phenotype differences?
Well, only looking at the responses, in majority of those switching neurons, we see this auto-inhibition after this excitatory component of response. So we can speculate that maybe this population has D2 receptors, auto-inhibitory receptors, but there's nothing more that we can gather from this electrophysiological signal.
Okay, thank you very much.